the interface between the nervous system, organ physiology, immunology, reproduction, aging. So stay tuned for updates in our website and worldwide neuro seminars. So before I introduce our today's wonderful speaker, let me remind you that you can ask any questions during the talk using the Ask a Question button. And also, if you want to meet the speaker after the talk, we will have a Zoom Q&A session that I will write the link in the chat in a bit. And now for real, I'm eager to introduce Dr. Sylvia Cremer. I am going to briefly summarize her impressive career, so we have time for her talk. Dr. Sylvia Cremer is a professor at the University of Science and Technology in Austria. She received her PhD degree at the University of, let's see if I say this right, Regensburg in Germany. After her postdoc at the University of Copenhagen in Denmark, she started her own research group in 2006. Her research group studies how collective protection arises at the colony level from individual behaviors and social interaction in ants. And more specifically, her lab focuses on the effects of pathogens on social interaction networks in ant societies and determines how interaction with sick nestmates affects both the hygiene behaviors and the physiological immune system of individual ant workers. In this sense, ant societies and the pathogens are an ideal model system to understand epidemiology in societies. So today she will be talking about the social immunity and the disease defense of the colony. So thank you so much, Sylvia, for being here today with us, and the stage is all yours.
Yeah, thank you very much, Raquel, for this introduction. I will make everybody else mute so that I also don't hear myself twice. So if you have any questions, I will only be able to hear them later on after the talk. So thank you very much again for the invitation. And I'm happy to talk to you about social immunity in ants. And basically how the whole colony is defending itself against disease. So, of course, the data that I'm going to show here have been work of a lot of people from my group. This is basically people that have been working with me in the past and then also with some collaborators outside of the group. So I don't think I need to go a lot into detail to tell you that in societies where a lot of hosts have a lot of very intense social contacts, infectious disease can easily spread between the hosts. And this is equally so in human societies as well as in the societies of social insects. And here they have a lot of very frequent and very intimate interactions. For example, social insects are sharing their food by regurgitation, and they constantly lick one another's bodies to exchange the cuticular hydrocarbon waxes of their exoskeleton to have a shared colony odor. So these social interactions that these ants are sharing in a colony, they can very easily promote disease transmission. But then, on the other hand, these social interactions can also help to reduce disease risk if it is hygiene behaviors that are performed together. So this brings me to the concept of social immunity. If you look at an individual ant in this case, this individual has its own individual immune system, and that comprises both its physiological immune system that also insects have, and they are relatively well in defending themselves against pathogens, as well as also an individual hygiene behavior that each colony member has. And if these different individuals are then cooperating with one another to perform hygiene collectively or towards one another, then the additional level of social immunity can arise from this. And social immunity is very good in reducing disease at different levels. On one hand, it can reduce the pathogen load of the colony. It can reduce the susceptibility of the hosts, as well as the disease transmission. And I will go through these different ways how social immunity is reducing disease. The model system that we work most with in my group is using ants as social insects, so social insect hosts, and these are typically garden ants and fungal pathogens that we can rear here on an agar plate, and the spores or conidiospores of which are then attaching to the body surface of their insect hosts, and they then start to grow into the host, cause the host death, and then form mycelia, and then again spore packages out of the dead host. And we have shown already that ants can defend themselves against diseases by, first of all, a very good level of sanitation or nest hygiene. If you see here an ant colony that has moved into a nest where we used a pH-sensitive paper as ground, we can visualize very nicely that the ants are spraying their own poison, which in these garden ants consists of formic acid, and then turns the pH-sensitive paper red in the areas where it has been sprayed. So we know that formic acid is good in preventing fungal and bacterial growth. So basically what the ants are doing is that they prophylactically already clean any new nest that they move in. In addition to nest hygiene, very general nest hygiene, what they also do is sanitary care. So that means when they interact, when they contact a pathogen-exposed nestmate, then they start allogrooming this individual. Allogrooming is a sanitary care action that has two components. The first, widely recognized one, is that the ants are using their mouth parts to remove, to more or less pluck off these external spores from the cuticle of the host. So they basically remove all the pathogens from the body surface of a contaminated nestmate. But in addition, what they also do is that they again use their antimicrobial poison, and in this case they don't necessarily spray it from their poison gland directly, but they take it up by bending their mouth to the abdominal tip, to their poison gland, take it up into the mouth, and then apply it during the grooming on the body surface. So this is a combination of mechanical removal and chemical disinfection. And this, as a lot of people have shown, is reducing the disease risk of these contaminated individuals. But we were very interested also on the effect on the nestmates themselves, because if you have very close contact to an infectious colony member, then there is a risk of pathogen transfer. So we wanted to know whether nestmates get an internal infection, and we did so by dissecting their body content and putting it on agar plates and then counting colony forming units. And of course, we first did this with the directly exposed individuals, where you can see that each agar plate is the body content of a single ant that we had directly exposed. And you can see that most of them have very high load of fungal growth on these agar plates. And now to the interesting question, how about the nestmates? And you can see here that also very many nestmates do have an infection, but much fewer colony forming units. So it seems that the majority of nestmate contracts what we would call a low-level infection. So if you compare now the directly exposed individuals and their nestmates, you can see that after having dipped all of these in fungal spores, 90% have developed an internal infection, whereas even 65% of the nestmates, so very many. But this was an LD50, so the exposed had a mortality of about 50%, whereas the nestmates, only 2% died. And that was because their infection load was about 12-fold lower than that of the directly exposed individuals. So this means that the majority of the caregivers, the nestmates, have been developing asymptomatic low-level infections that didn't cause disease. Instead, what we could see is that these nestmates actually upregulated their antifungal immune genes, which I don't show the data here now, but this then led to an increased immune function. And here you can see the inhibitory capacity of the body content of these ants. And these are the baseline ants of sham control, so healthy ants, whereas these low-level infected individuals, they actually have a higher ability to decrease the pathogen load. And this then also relates to a survival benefit of these nestmates after own challenge. So if you have nestmates that have only lived together with other healthy individuals, the blue line, and you expose these to a high fungal load afterwards, then they have a much lower survival than those nestmates that have received a low-level infection from having lived together with a highly exposed individual. So what we could say is that these nestmates of exposed individuals receive kind of a protective immunization that protects them against a disease later on. So yes, indeed, we can say that there is pathogen transfer, but that doesn't lead to disease in most of the cases, but to a protective immunization. So the protective effect of low-level infections against future disease is what we can observe naturally in the ants. And it has been made use of already in very early human medicine before Jenner invented modern vaccination as what has been named variolation or inoculation against, for example, smallpox disease. So if then a great proportion of the colony receives this immunization, then the colony can actually receive herd immunity. And this is then, of course, reducing the or altering the colony transmission dynamics. But we were also interested whether it also affects the performance of individuals to then perform sanitary care to other contagious individuals, depending on their own immune state. So basically what we did is that we had nestmates that we let interact with the pathogen-exposed individuals so that they received a low-level infection. And then we introduced a new individual to those nestmates that either carried the same disease or a different pathogen. And we know already that the nestmates are protected against this homologous fungus, whereas we also knew that they became very susceptible to this heterologous fungus because superinfections are actually something that can be very dangerous, also in the ants. When they have a low-level infection with one fungus, in this case it's a combination of Metarhizium and Beauveria insect fungi, in either combination, Metarhizium Beauveria or Beauveria Metarhizium first, then these nestmates are highly susceptible to this second heterologous fungus. So in our experiment, we then wanted to test whether these low-level infected ants are avoiding these contagious colony members to prevent a harmful superinfection with the heterologous pathogen. And we observed the behavior of the ants, and we observed actually aggression, which is extremely rare to observe in ant colonies, as well as sanitary care. So in these next slides, I will always show you the non-infected control ants, and when the second incoming ant either carried this Metarhizium fungus in black or this gray Beauveria fungus. And so these individuals had been sham low-level infected, so they didn't carry any infection. And in this case, they had a homologous infection, so in this case also Metarhizium or also Beauveria. And here they had the heterologous low-level infection, so a Beauveria and a Metarhizium infection. And what you can see here is that there is aggression occurring. The ants are starting to bite a bit and drag along these contaminated second incoming individuals. And we found that these low-level infected ants, they showed higher aggression towards these contagious nestmates than non-infected individuals. Now, aggression is a relatively typical sickness behavior, and we could now also find that it can occur in ants. Mostly, we found that the low-level infected individuals are still performing sanitary care. And as you know, sanitary care is grooming plus then also poison spraying or poison application. And what we found, if we look at allogrooming, we could confirm previous work by other people that have shown that if an individual has had previous contact to a pathogen, then it is actually becoming more alert and starts increasing its allogrooming levels. So this is what you can see here, that there's an increase in grooming frequency from the baseline of non-infected individuals to this homologous low-level infected ants. And interestingly, what we found now is that this was actually not the case. So there was actually a reduction in grooming again in the heterologous combinations. So even if they are typically more alert, they kind of shy away from grooming those individuals that carry the, for them, very risky second pathogen. And if we look at the poison spraying, then we actually found the opposite, namely that they are spraying a lot against these heterologous contaminated, contagious individuals. So low-level infected ants groom less, but specifically disinfect those colony members that are contagious with this harmful pathogen to themselves. So these behavioral changes in the ants, they then led to a reduced superinfection level. So that means the number of spores that they contract during the social interactions. This number was relatively high in both the non-infected and the homologously infected ants, but it was drastically reduced in the heterologous situation. So these low-level infected ants acquire much less viable pathogen and thus develop lower superinfection loads in the heterologous case. So this means that the ants, even if they are if they are facing a colony member that carries a pathogen that is very harmful to themselves, they do not stop taking care of this individual. They are still providing sanitary care, but they perform it in a way that is protecting themselves from receiving receiving a secondary infection. So they perform risk-adjusted sanitary care by modulating the two types how they are performing sanitary care. They spray more and they groom less. So this means that susceptibility-driven changes in sanitary care are leading then to reduced disease transmission in the colony. And we also looked at social distancing in whole colony setups upon pathogen exposure. And to do this, we teamed up with the group of Laura Keller at the University of Lausanne that had developed an automated tracking system that you can see here. You can glue a QR code on each individual end of the colony. So these are the workers, and this is the queen, and of course the brood you cannot tag, but it's also not moving around. And then this is their technical setup, and with this what you can do is that you make 2 images per second to receive the position and orientation of any individual in the colony. So this is how this looks like for our experiment. We here have a nest, and you can see all the ants that have been detected automatically by the system. They receive a red dot and a number. There are a few that have not been detected, probably because the tag was a bit bent at this particular frame. And this is the outside nest area, the foraging area, where we provide water and sugar to the ants. And these individuals that are leaving the nest, they are called forager individuals. So what you can then do based on how these tags, the XY positions over time of these tags are changing, for each of the ant you have some kind of like this body area, and you can then see if these 2 body areas are overlapping, and you can infer that there has been an interaction going on. It's and you can form a proximity network based on all these automatically taken informations. So by doing that, what we could find is, and here again we could confirm previous work, is that the interaction network of a social insect colony is not homogeneous. But what happens is that there are subgroups. Here you can find the purple queen, and you have the green nurse workers. And in yellow, you find those individuals that are also leaving the nest and are the forager individuals. And you can already see that the foragers are interacting more with one another than towards the intranest workers. The individuals that carry the black dot here are the ones that we later in the experiment then exposed to the fungal pathogen. And this is how then the network changed after this treatment. What you can see here is that the already existing subgroup formation has become stronger. And this is because of behavioral changes. And you can see here those treated foragers that we had exposed, they spent much more time outside the colony. But what was very interesting is that also the untreated foragers, so their colleagues that are sharing the same task with them, they also spent more time outside, whereas the nurses didn't change the behavior, spatial usage. Similarly, if you look at the distance to the colony in terms of network analysis, the same was found. Treated individuals distance themselves, untreated as well, and nurses don't change. So there is a behavioral modulation of the exposed ants plus their healthy peer group. And this then has an effect on the individual pathogen load that we measured for each individual at the end of the experiment. So we could see here that, of course, we had applied the pathogen to the treated foragers. So we here only look at the untreated foragers as well as the nurses and the queen. And you can see here that the queen and the nurses have a significantly reduced probability of receiving that high load that leads to disease and death. And on the contrary, they have a significantly increased probability of receiving a low load. And as we have heard before, this low load can lead to some immunization. So basically, you have a double advantage, so to speak, as a nurse and queen in these colonies, you have a lower probability of receiving a disease and a higher probability of getting kind of vaccinated or immunized. So with this work, we could also not only establish that the ants show social distancing, but also that the social distancing is then leading to a reduced pathogen transfer. So this means that social immunity does reduce disease. First of all, like a resistance mechanism, it is reducing the pathogen load by sanitation. It is reducing the host susceptibility by social immunization, and it is decreasing the disease transmission due to the fact that the sanitary care is performed in a risk-adjusted manner, as well as that the ants are showing social distancing. So we also wanted to know whether these sophisticated social immunity measures have an effect on the pathogen community that they co-evolve with. And just to show you here again these two pathogens, Metarhizium and Beauveria, there is a high pathogen diversity even within these entomopathogenic fungi that build these spores that are in the soil, and there is a lot of spores in even like 5,000 spores in 1 gram of soil that comprise very many different strains and species of fungi. So we wanted to know whether this social immunity does shape the pathogen community. And for this, we performed a co-infection experiment or a co-exposure experiment where we let different fungal pathogen species compete for the host, mimicking the natural situation where spores of multiple species are then attaching to the host cuticle. And what we found is that those species where the spores are actually germinating faster and are then penetrating into the host body at a faster speed, that these are exposed to the allogrooming by the nestmates for a shorter time period, which is then increasing their chance of competing successfully with the other pathogens and to then grow out spores themselves out of the dead host. So we made a model, or basically Christian Hilbe made a model for us to compare the dynamics under individual host context and this group host context, so without and with social immunity. And the yellow line here is the fast germinating species. And what you can see here is that the pure fact of the increased germination speed of this species plus the allogrooming of the hosts that is present here but not here is leading to a success of this fast germinating fungus. So social immunity modulates pathogen competition, and this is something very similar to our own body, where our immune system is also creating a situation that can favor or disfavor one or another pathogen when we also have co-infections of multiple pathogens that are competing in our body for our host resources. So together, this means that social immunity does change disease ecology. And with this, I'm at the end of my talk, and I'm happy to take your questions. And for this, I will try to hear you again. And maybe I can even see something.
Okay,
I can hear you,
now?
yeah.
Perfect. That was really, really nice. This is very, very impressive. Wow. Thank you so much. So yeah, if anyone in the audience wants to post any questions, they can do it using the Ask a Question button, and I can read it out loud for you. So, well, I can start just because I think this is really, really fascinating. So for this to happen, first there's need to be like a recognition part from the ant that it's infected, and then it needs to trigger anything that is, even if it's the nurse, or I don't know if also the infected ant can clean itself by grooming and all that. So it needs to recognize a pathogen, induce the behavior, induce all this immune system. Is it known how, like, molecules or the circuit that all these, how this all is
I
happening?
don't think it is very well understood. What we think is that there is some olfaction involved, because the very interesting point is that the ants can realize the pathogen itself. They react immediately and they don't have to wait for symptoms to develop. So there are some ideas that, or I think there has been some work found in Drosophila, so that there is some olfactory receptors that can then link directly to hormones and to changes in the immune system. But I don't think there's anything really known in the social insect.
I see, I see. Okay, so, well, first, Anastasia is saying thank you for your nice presentation. I think you're seeing it there. And Jerica is also saying fascinating talk. Sorry if I missed this in the beginning, but is it known how the ants are sensing pathogens and how they differentiate pathogens?
No, not that I know. This is a field of chemical ecology, and this is something that people start becoming quite interested. We also are trying to solve this. So the problem that the ants are facing multiple problems, because there is a lot of microbes in the environment and not all of them are pathogens. So the question is, how also does this become specific? And we are not even sure how specific the behavior is. My feeling is that mostly the ants are performing a very general hygiene against mostly everything that touches them. Yeah.
Okay, I'm starting to hear a little bit of background noise. I don't know if it's me, the only one, but in any case, we have another question. I could still hear you pretty okay. We have another question from Lina. Thank you for a wonderful talk. I'm interested in the increased aggression towards sick individuals. Will this not increase the pathogen exposure?
Okay, so the aggression was very rare, and I had the feeling that what the ants wanted to do is that they drag out the other individual, maybe from the central core of the nest, and they rather did this by grabbing a leg and then dragging this individual. So I don't think there was a very close social interaction that would lead to a high transfer of the pathogen.
Yeah, I think that was actually a good point. And is this because we have this example in ants? Is there any vertebrate example that these kinds of things happen? I mean, besides I see someone sneezing and now I just want to run away, but I don't know if I will be able to detect that someone is infected without, like, symptoms. Is this the same for, I guess you said that for ants is different because they don't need to get sick to be recognized as infected?
Okay, maybe one point that was also asked before, this contaminated individual itself is also grooming itself, which is called self-grooming. And so it shows a behavioral change, and we don't know whether the nestmates may also react to this behavioral change of the contaminated individual itself. So it can be olfaction, but also reaction to behavioral changes in these exposed individuals. There is quite a lot of literature that there can be some avoidance, also in other insects, towards contaminated sites, or also to infected individuals, also in other invertebrates. For example, lobsters also don't want to share hiding under a stone with an infectious individual. And I think that mostly people assume that it must be some kind of olfaction or taste receptors. And there is interesting work in Drosophila that also shows that I think some olfactory receptors or taste or gustatory receptors, even in the wings, if I'm not mistaken, are inducing self-grooming of the flies. So there must be a clear link somehow between olfaction, immune system, and behavior that I don't think is very clearly understood at the moment, or at least not in this audience.
Okay, I see. Very interesting. Thank you. I don't know if anyone else has more questions. Now is the moment to ask. Otherwise, I think that I'm going to thanks once more, Sylvia, for today's presentation, and also everyone for joining today. Our next speaker is going to be Tony Wiscorey next Tuesday, and he's going to talk about systemic regulation and measurement of mammalian aging. And actually, that is going to be our last seminar for this season. So I hope to see you all there next week. And for now, everyone who wants to join, I posted here the Zoom link. So if you want to join the further discussion with the speaker, that will not be recorded. So come on. See you. Bye bye.
Yeah, okay.